Hello, welcome to Biology, the scientific study of life. Reproductive cycles of phages. Phages are the best understood of all viruses, although some of them are also among the most complex. Research on phages led to the discovery that some double-stranded DNA viruses can reproduce by two alternative mechanisms, the lytic cycle and the lysogenic cycle. Let's begin with the lytic cycle, a phage reproductive cycle that culminates in death of the host cell is known as a lytic cycle. The term refers to the last stage of infection during which the bacterium lysis breaks open and releases the phages that were produced within the cell. Each of these phages can then infect a healthy cell, and few successive lytic cycles can destroy an entire bacterial population in just a few hours. A phage that reproduces only by a lytic cycle is a virulent phage. Illustrates the major steps in the lytic cycle of T4, a typical virulent phage. This figure, notice that and legend describe the process. The lytic cycle of phage T4, a virulent phage. Phage T4 has about 100 genes that which are transcribed and translated using the host cell's machinery. One of the first phage genes translated after the viral DNA enters the host cell calls for an enzyme that degrades the host cell's DNA. Steps 2. The phage DNA is protected from breakdown because it contains a modified form of cytosine that is not recognized by an enzyme. The entire lytic cycle from the phages first contact with the cell surface to cell lysis takes only 20 to 30 minutes at 37 Celsius degree. Notice here the phage assembly. Notice this one represents head, and in the middle these tails, and these lines tail fibers. Okay. Attachment. Let's begin with the steps. Also, attachment. First steps is attachment. The T4 phage uses its tail fibers to bind to specific receptor sites on the outer surface of an E. coli cell. Notice too also entry of phage DNA and degradation of host DNA. The sheath of the tail contracts, injecting the phage DNA into the cell and leaving an empty capsid outside the cell's DNA is hydrolyzed. Steps 3. Synthesis of viral genomes and proteins. The phage DNA directs production of phage proteins and copies the phage genome by host enzymes using components within the cell. Third, assembly three separate sets of proteins, self-assembly, to form phage heads, tails, and tail fibers. The phage genome is packaged inside the capsid as a head forms. 5. And last one, last step also release. The phage directs production of an enzyme that damages the bacterial cell wall, allowing fluid to enter. The cell swells and finally bursts, releasing 100 to 200 phages partials. Okay, notice after knowing that, or notice that this represents the lytic cycle. We might wonder why phages haven't exterminated all bacteria. In fact, phages or phage treatments have been used medically in some countries to help control bacterial infections. But bacteria are not defenseless. First, natural selection favors bacterial mutants with receptor sites that are no longer recognized by a particular type of phage. Second, when phage DNA successfully enters a bacterium, the DNA often is recognized as foreign and cut up by cellular enzymes called restriction endonucleases, or simply restriction enzymes. The bacterial cell's own DNA is chemically modified in a way that prevents attack by restriction enzymes, but just as natural selection favors bacteria with effective restriction enzymes, natural selection favors phage mutants that are resistant to those enzymes. The parasite host relationship is in constant evolutionary flux. There is yet a third important reason bacteria have been spared from extinction as a result of phage activity. Instead of lysing their host cells, many phages coexist with them in what is called the lysogenic cycle. Lysogenic cycle. In contrast to the lytic cycle, which kills the host cell, the lysogenic cycle replicates the phage genome without destroying the host. Phage is capable of using both modes of reproducing within a bacterium are called temperate phages. A temperate phage called limpido, written with the Greek letter A, is widely used in biological research. Phage A resembles T4, but it still has only one short tail fiber. Infection of an E. coli cell by phage E begins when the phage binds to the surface of the cell and injects its DNA. Within the host, the eDNA molecule forms a circle. What happens next depends on the reproductive mode, lytic cycle or lysogenic cycle. During a lytic cycle, the viral genes immediately turn the host cell into a producing factory, and the cell soon lyses and releases its viral products. During a lysogenic cycle, however, the DNA molecule is incorporated by genetic recombination, crossing over into a specific site on the host cell's chromosome. When integrated into a bacterial chromosome in this way, the viral DNA is known as a prophage. One prophage gene codes for a protein that prevents the transcription of most of the other prophage genes. Thus, the phage genome is mostly silent within the bacterium every time the E. coli cell prepares to divide it. It replicates the phage DNA along with its own and passes the copies onto daughter cells. A single infected cell can quickly give rise to a large population of bacteria carrying the virus in a prophage form. This mechanism enables virus to propagate without killing the host cells in which they depend. The term lysogenic implies that prophages are capable of giving rise to active phages that lyse their host cells. This occurs when the A genome exists, the bacterial chromosome, and initiates a lytic cycle. What triggers the switch over from the lysogenic to the lytic mode is usually an environmental signal, such as radiation or the presence of certain chemicals. In addition to the gene for the transcription preventing protein, a few other prophage genes may also be expressed during lysogenic cycles. Expression of these genes may alter the host's phenotype, a phenomenon that can have important medical significance. For example, the bacteria that cause the human diseases, diphtheria, disease such as diphtheria, botulism, and scarlet fever would be harmful to humans if it were not for certain prophage genes that cause the host bacteria to make toxins. Reproductive cycles of animals or animal viruses. Everyone has suffered from viral infections.